Hello everyone and welcome back to our show. Today we will be discussing the topic of friendly phages find foes. To preference this, we first need to know what exactly a phage is. A phage is a virus that infects a bacteria and then divides within them. And another important mention of a phage is that it also helps to distinguish between different types of bacteria. The difference between two types of bacteria is what phages they carry. Another mention is that there are two types of phages, a lytic phage and a lysogenic phage. The lytic phage reproduces within the cell and destroys the cell. This kills the cell immediately. A lysogenic phage inserts their genome and divide with the cell. This causes the cell to become stressed. So basically, a lysogenic phage actively or silently divides with the cell, and eventually they will take out their genome and go and cause stress to other cells. Okay. So, as you can see here, there are two different types of bacteria represented. Each bacteria has what is called a flagellum on the end of it, and these flagellum help the bacteria to move. And how the bacteria interact with each other is through a chemical process. They they send out a certain type of molecule in which the concentration of this molecule reaches what it needs to be. The bacteria will change accordingly and they will change together. So scientists were conducting a study and they realized that when you have two different types of bacteria, they will not interact with each other. Instead, at the midway point, there will be this line of demarcation. And this line of demarcation only appears when you have two different types of bacteria, because they saw that when you have one type of bacteria but two different types of colonies together, they will interact and they will cause no line of demarcation. And so this made scientists wonder what type of gene could be causing this line of demarcation. What type of gene could be causing this line of demarcation? And to figure this out, they actually took various strands of E. coli, and they would go through and they would knock out one gene in each strand to determine what gene was causing. This resulted in 8 knockout genes showing a lighter or weaker line of demarcation. And this also led to one knockout gene resulting with no line of demarcation. So this helped narrow down that REP, which is the replication of prophages, was a playing factor in what was causing this non-interaction in this line of demarcation. And prophages are lysogenic phages that insert themselves into genomes. And another factor was SW1. And SW1 is a lytic phage that is dependent on the genes of the prophage to help it reproduce. So as you can see here, scientists have now concluded that lysogenic and lytic phages are a playing factor in why this line of demarcation is. And let's see. Oh, and to mention that the REP, the prophages, the function is not well known, but it's important for phages to kill bacteria. Which seemed to be the reason for the line of demarcation. So this is why REP was causing this. Okay. So when you delete all of the phages that were represented in REP, when you delete all 9 phages, there was no line of demarcation, as you can see here, and the bacteria would interact with each other, and there would be no visible line. So, and what they found from this was that there was one type of phage causing it, and the genes that they were carrying, there were 2 genes, 2 viral genes that was causing this non-interaction to occur. So the scientists figured that the lytic phages, let's go back to this definition. The lytic phages would sit within the cell, and they would ride on the edge, and as they would approach a non-carrying cell, non-carrying of the specific phage, that they would, when they would get close, they would attack and destroy those phages. So the carrying phages were seen as the carriers, and the non-carriers were seen as non-carriers. Non-carrying of the phage was seen as non-carriers. And this is ironic because, as we can see here, lytic phages are supposed to kill the cell immediately. So this was ironic for the scientists to find that this was actually what was occurring. And it was also determined that different mixtures of different phages also seem to have the same effect. You basically just needed a resistant and a non-resistant phages. So the carriers were the resistant, and the non-carriers were the non-resistant. And the carriers were seen as the more fit phages, because they would, when they would attack the non-resistant, they would get all the nutrients, and sometimes in some cases they would even destroy. So they were seen as more fit because they were getting more nutrients and killing. And they would drive back the non-carriers, and this would be the reason for the line of demarcation. But it does, however, cause for this process to happen. You obviously have to produce the phages, and you have to have the protein involved to drive the process, to carry out the process. And then for this process to be useful, the virus has to have its own type of immunity. If there's no enemy around any of the no non-resistant or no enemy around the carriers or the non-resistant, then they would end up killing their cells and destroying their cells, and the process would be non-applicable. So, in conclusion to this, this process can be important for when analyzing our guts, because obviously we have bacteria, land, bacteria in land, water, any type of water, and this can be applied to the prevention of colonization and diseases, because obviously these carriers would push back the non-carriers, preventing colonization. This process may also be used to improve phage therapy, which is to prevent phages from gaining further immunity to other pathogens. To go back and explain exactly what this title means, it says friendly phages find foes. As you can see here, a phage is a virus, and you think, how can a phage be friendly and how can a phage be helpful? But basically, not all viruses are harmful, and not all phages are harmful, because they are pushing back these non-carriers that may potentially be harmful to the human or any type of health.